I'm Carika Weldon, a scientific researcher at the University of Oxford, and I'm here today with Rando from UMass Medical School, and welcome.
Thank you. Thanks for having me.
Thank you for coming. We're at the 84th Cold Spring Harbor Symposium on RNA control and regulation. You had a stellar talk yesterday. You captivated everyone's attention the whole time, obviously with your great science, but also with your charisma.
Why, thank you.
You're welcome. So can you give us a brief kind of summary of what your work was about that you talked about yesterday?
Sure. So the general area we work in is paternal effects in mammals, so effects of a male's environment on his offspring. And so we spend a lot of time working on different things that you can poke dads with that'll affect metabolism in kids or anxiety-related behaviors. And most of what I talk about these days at meetings like this is our attempts to understand how this works. So how what a dad sees or experiences affects information in sperm, how sperm information affects early development, and how that might ultimately give rise to a phenotype in the kids.
And I remember very clearly that your title caught everyone's attention.
Daddy issues.
It started with daddy issues. I was like, whoa, it's got a bit deep all of a sudden.
Yes.
But you definitely lightened the mood. Okay, so if you want to dig a bit deeper into how exactly does the father's environment affect the child?
Right. So there are at least 3 different kinds of epigenetic information that make it into germ cells. There are small RNAs, there's DNA modification like cytosine methylation, and then there are histone modifications. And so we think in our system, so the system we mostly focus on is dads eating a low-protein diet.
Okay.
And in that system, we don't think there are many reproducible effects on DNA modification or on chromatin. So we are focused on small RNAs. And so we don't know for sure that small RNAs in sperm are responsible for programming offspring, although there are several other studies that have reported microinjecting sperm RNAs and getting phenotypes in kids. We haven't tried that yet, but we've focused very much on characterizing the small RNAs that are in sperm and trying to figure out where they come from and what they do when they're in the embryo.
I just want to clarify for the audience listening, this is not in humans, right?
We work in mouse.
All right, because there was one point during your talk and I was like, wait, what is he testing this on? Because you're like, yeah, we just took it and then we just injected it. I was like, wait, but it is on animals.
That's right.
Right? Just to clarify anyone's, questions out there. Awesome. Okay, I've always heard that the age of the father actually affects the child. I've never heard the diet of the father affects the child. Is that independent of age, or
certainly in the way that we and other people do experiments, it's independent of age, because you take an animal on control diet who's 12 weeks old and an animal on high fat or low protein diet who's 12 weeks old. So it's not like you're taking a 2-year-old high fat animal and a 12-week-old control. So the age is controlled for. There is plenty of evidence that paternal age affects, in humans, it's correlated with autism and schizophrenia, I believe, in the next generation. But we don't study that. Those are long-term experiments.
Yeah. Okay, so looking at the diet, you're saying that if the father has a low-protein diet, that then is a negative for the child?
Well, okay, so in terms of whether it's useful or not, we don't know. So what it affects in kids is it alters the levels of, we think, bile acids. So it affects the levels of hepatic cholesterol esters.
Okay.
And it affects the levels of hepatic regulation of cholesterol biosynthesis genes. It affects glucose control. So all of those things can be good or bad depending on whether or not the kid is seeing a scarce diet or a plentiful diet. So the system I didn't talk about, but I think is really useful for thinking about, like, is it good or is it bad? So we also study paternal nicotine.
Okay.
Kids whose fathers consumed nicotine are resistant to an LD50 injection, so a lethal injection of nicotine. So dads experiencing nicotine makes kids nicotine resistant. So this is good if you're going to be injected with an LD50 of nicotine as a child. But those kids are also bad at glucose clearance.
Okay.
So calling it good or bad, it's a trade-off, right? So it's good if you think the kids are going to be seeing toxic levels of drugs in the environment. It's bad if you think that they're not going to be and they're trying not to get diabetes and metabolic syndrome.
Right. Okay, so this I think goes to like the age-old question. I think you touched on it yesterday, that the sperm is not just delivering half of the chromosomes and then gets disintegrated and that's it. There's actually a bit more to what the sperm is delivering, because most people, I think in this age, you say, you know, the egg is everything. It's just, you know, the sperm is just a little part of it. But actually, no, it's a bit more.
No, it's Dad's fault too.
Yeah. I like that. So I want to know, how did you get into this field? Because it's not your typical field that I've heard about at an RNA meeting. So it's very interesting for me to know, you know, how did you pick this field?
So I started in chromatin and transcriptional control. And one of the interesting things about chromatin is that it, at least in some cases, seems to carry epigenetic information at cell division. And just I was just starting my lab, and I was thinking about interesting things that epigenetics could do. And one of the things people talk about a lot, but hadn't tested very much, because epigenetic information often listens to environmental conditions. So like chromatin state changes when you heat shock yeast or change their conditions. If any of this makes it through germ cells, that would allow the environment in one generation to affect the future.
Right.
And it was the kind of thing that people had started to talk about a lot, speculatively, right? Because it's reviving a very old question.
Yeah.
So we just set out to test it, and it took a really long time to figure out a useful system for it and stuff like that. But
okay. And how about your colleagues? How did they receive what you do in terms of your research?
Well, you probably have to ask them. I'm sure there's a lot of, I know there's a lot of skepticism. People don't express their skepticism to me personally that often. And when they do, it always hurts, but I try to take it positively.
Yeah.
But, you know, at this point, simply the observation that a paternal diet affects offspring metabolism has now been sort of repeated in various ways in something like 100 publications. So I think just by sort of sheer mass of publication, people are starting to believe that idea.
Yeah.
I certainly don't believe that we know the mechanisms by which this works in any great detail. And so as to whether or not the mechanisms we're working on are going to ultimately be the correct answer for how dads talk to kids, I myself am skeptical.
Yeah.
I think some people believe that, you know, the small RNAs we work on might be the answer, and I'm sure lots of people don't. And I don't know which of those people is correct.
Well, time will tell, right?
I hope so.
I want to touch on a more global aspect of science. You mentioned that, you know, there's some skepticism. And that happens in, I'm pretty sure, every area of science. And it also happens when there's like a big shift in the thinking, when there's a result that doesn't fit with what everyone thinks. Well, what do you want to say about that in terms of, you know, in general in science, how we should handle change and how we should handle not ignoring results because it doesn't fit what we think?
Well, I mean, I don't have anything super broad or useful to say about that. I mean, I think the point of science is that if evidence forces you to discard a long and well-loved hypothesis, the whole point of the scientific method is that you have to listen to that evidence. So I think most people ultimately will sort of come around. The flip side of that is that, you know, if people have believed something for 100 years, then extraordinary claims require extraordinary evidence. So I think it's very healthy to sort of demand a high level of evidence for something that people suspected wasn't true, right?
Yeah.
So I think that, you know, extreme skepticism is warranted and healthy.
Yes, yes.
But on the other hand, it's also the case that ultimately, if, you know, when the preponderance of evidence argues against your deeply held beliefs, you should be really paying attention to the evidence.
It's time to let it go.
Yeah.
Yeah. Okay, there's another thing I wanted to ask you. Flip side of the science, your charisma. I mean, like I said, that captivated a lot of people yesterday. I found this topic initially when I read the abstract. I was like, this is very, this is very out there for me. But you made it so easy to accept and so easy to listen to and to understand. How do you find that as a scientist in terms of delivery of the science? How do you find that balance?
Well, okay, so one thing, I have no idea if I'm charismatic or not.
You are.
I do know that it's easy to talk about reproduction because
Okay.
everybody can relate to reproductive biology.
Yeah.
One way or another. So that makes it really easy. I also personally am I'm quite shy, actually, and so as a result, I have to give a talk. I have to sort of like it's like acting. You know, I have to feel like it's a performance. So I think that's part of why I'm very sort of like throwing my hands around and making lots of jokes. Just sort of like keeps me comfortable. But I think it's absolutely. I mean, it is the case that there are people who do amazing science who I just can't watch their talks. And I get nothing out of their talks, and I'll just, you know, I'd be better off reading papers than seeing the talks. So the delivery means a lot, obviously, because sometimes you don't realize how special a body of work is if people can't communicate that to you. So science communication is, you know, super important for influencing how people think about the world.
I agree. I think you're able to captivate a new audience, and you're able to, you know, get people excited about what you're doing.
Yeah.
And that also is very important because then people can buy into it. Back to the science, you talked about two RNAs that for me, I mean, I work on RNA splicing, so mRNAs is kind of just what I talk about. We talked about piRNAs and then these tRNA fragments, so TERFs,
Yes.
if I remember correctly. Can you elaborate on those a bit more?
Yeah, so people are generally familiar with, in germline, the piRNAs are a pretty famous class of small RNAs. They're an well, it depends on the organism, but one of the roles they play is an anti-transposon defense system. tRNA fragments are what's unusual, sorry, I'm going to start that over. Sperm are unusual in the RNAs they carry because unlike most cells of the body, they carry some microRNAs, which is the most common small RNA in a typical fibroblast or hepatocyte or something. But they carry much more of these tRNA fragments, which are the 5' ends of mature tRNAs. And those two statements, or those two stories, piRNAs are famous germline RNAs and sperm carry tRNA fragments, is sort of is a weird what's the word I'm looking for? It's not a paradox, but you think of the RNAs in the male germ cells as piRNAs because the testis is filled with them, but an ejaculated sperm carries totally different RNAs.
Right, okay.
And so that distinction actually led us to figure out where sperm RNAs come from. So they actually aren't made during the process of sperm development in the testis. They're shipped into sperm as they spend a week or 2 weeks going through this long tube called the epididymis.
Okay. And if I remember correctly, you said that is one part of the body that does not get cancer?
That's what I've heard. And pathologists when I sort of say that at talks, pathologists will sort of come up to me afterwards and say, yeah, you know, I looked it up and there are like no reports of epididymal cancer.
Do you think that's linked to the small RNA production maybe?
It's an interesting idea. We don't study cell proliferation, but it certainly bears looking into.
So okay, let's recap again. So you're saying that in the testes, all of the RNA mainly is piRNAs, but by the time it becomes mature and is released as sperm, it's not piRNAs.
That's right. So as sperm leave the testis, they eliminate somehow, whether they're degrading them or dumping them into a blobby, membrane-bound sac. They eliminate all their piRNAs, and as soon as we can get them, so in the very early epididymis, they've already gained all tRNA fragments.
Do you know how that happens? Or?
We think the way they gain tRNA fragments, so we don't know anything about how they lose piRNAs, sort of assume it's degradation. The way they gain tRNA fragments is the epithelium of this tube makes tRNA fragments, and we believe it ships them over in little vesicles called epididymosomes.
Interesting. Okay, so it's swimming. Is it swimming?
They don't know how to swim yet.
They don't know how to swim yet.
One of the main things they learn in the epididymis is how to swim.
Okay, so they're kind of like just moving along,
getting pushed by fluid flow, riding the
wave.
Yep.
They're riding the wave down the epididymis, and they're along the way gaining these new small RNAs, which are the TERFs, the tRNA fragments.
Correct.
And then, if I remember correctly, you had, right before they were ejaculated, they did gain back some RNAs, or
No,
it was like a dip. I remember that.
Right. So then the third class of small RNAs in the picture are the microRNAs.
Okay, yes.
And there are a small subset of microRNAs that are present in testicular sperm, absent in sperm in the early epididymis, and then present again in sperm in the late epididymis, which is really weird.
Yeah.
Whether or not sperm are dumping these RNAs in the early epididymis and then regaining them later, or whether or not a mixture of sperm comes out of the testis and the ones who are lacking these RNAs are getting stuck in the early epididymis.
Right, okay, that makes sense.
Those are the sort of two models for it. But if you just purify sperm from the early and late epididymis, they differ mostly in the levels of a couple microRNAs.
Okay, so in terms of your field at the minute, in terms of this developmental change in the RNAs, where do you see this going? Is it going towards therapy, or what's the kind of like end goal?
Well, so I mean, our interest in it in terms of understanding how dads talk to kids. So our model is that the epididymis ships RNA into sperm that will be delivered to the embryo.
Right.
And we also know that the conditions that a dad lives in affect the RNAs that are in sperm. And so putting those together, our model is that the epididymis senses the dad's environment somehow and changes the RNA it makes to give to sperm, to give to kids. And so, among the million-dollar questions are, how does the epididymis see the world? How does its sensing of the world, of amino acid levels or of leptin or insulin or something like this, affect which RNAs it makes? So that's a sort of big basic question. You asked the question of whether or not this is something that one might manipulate to clinical ends.
Yes.
It's an interesting idea. I think it sort of runs into many of the same concerns people might have with editing the genomes of kids.
Okay.
So this would be editing the epigenome of kids.
Right,
and, that's above my pay grade in terms of discussing whether society wants to do it or not. But it's certainly, I think, at least reasonable to test the idea that by changing the RNAs you deliver to sperm, you would alter phenotypes in kids.
To me, this is really fascinating. I know that recently I had done like a school tour in my country, and we asked children to ask me questions about genetics. Now, each child that had the best question got a prize, and I actually went to my old school, and the child that got the best prize had a really interesting question. I think it actually links to your work. So the question was, you learn that you're 50% of your mom, you're 50% of your dad. He asked, can you be more of one than the other? What do you think? Based on what you know on your work.
Well, so let's see. I mean, generally speaking, we think of moms having more influence over kids than dads. And that's certainly true because in mammals, babies grow inside of mom.
Of course.
So, fetal alcohol syndrome is not magic and epigenetic, you're poisoning a baby.
Right.
And women with diabetes have so-called macrosomic kids. So they're just like stewing in sugar and they get huge.
Okay.
So in that sense, moms have a huge impact on kids that dads can't hope to replicate. In terms of the molecules in the germ cells, of course, oocytes are where you inherit mitochondria from. Sperm are where you inherit Y chromosomes from. But then beyond that, in terms of the small RNAs and the DNA modifications and stuff, it's still the case that probably more of what you inherit comes from the oocyte. Because generally speaking, mom tries to erase as much of dad's information as possible, so he doesn't make his kids get all the goodies.
Oh God, okay, that probably has a lot of implications in terms of society.
Yes.
That can be interpreted in many ways.
Yeah.
Very many ways.
That's probably not the best way to have phrased it.
No,
yeah.
No, it's fine. And this is the thing about science. I mean, you're seeing these results, right? Most likely, as you said, when this RNA gets injected into the egg is like, enemy, this is not what we want, get rid of it. But it's literally getting rid of information that would have went to the child. So, I mean, you can't rephrase it, can you?
Yeah, so I guess the assumption until like 10, 15 years ago was that everything except for half a genome came from mom.
Right.
And I think that assumption is now 90% true.
Okay.
So most information is probably still coming through the oocyte, but it turns out that at least some small amount of information comes through dad. And one of the questions we're quite interested in is what the bandwidth of sperm is. So exactly how much information does dad manage to get through the mom's screen?
Right.
Does he just tell kids quality of life? Life was crappy or great? Does he tell his kids about psychic stress and water supply and nutrition? Or does he tell his kids extremely detailed information about the world he lived in? And this, I think, is a really deep question for the paternal effect field, because while we know some information makes it through, the question of whether or not it's sort of coarse-grained information or very fine resolution information they have very different implications, right?
Of course. Okay, the last question I have for you is, where do you see this going in the next 10 to 15 years?
Well, so the big black box, so I'm very interested in how the epididymis sees the world and changes the RNAs it makes. In terms of our own work, the big black box is we now see changes in gene control in the early embryo, depending on which RNAs you give to the zygote. And then we know there are phenotypes when you have a pup. How changes in gene regulation in an early embryo turn into a cholesterol phenotype in the pup is it's a question that we have ideas about, but we haven't addressed at all. And I think that's the black box that the next decade or two is going to have to take seriously.
Thank you so much for your interview today. I really enjoyed talking about your work.
I enjoyed talking to you. Thank you.
Thank you.